So remember I said that translation is when we translate languages. We move from the language of nucleotides to the language of amino acids. And just like any translation, you need a decoder. And that's what the genetic code is. It's the way that the sequence of nucleotides in the mRNA match to a protein. So the genetic code is this chart that has been created by scientists, and it shows that a UUU, for example, so that's 3 nucleotides called a codon. So a grouping of 3 nucleotides is a codon. If that codon is in the mRNA, then in the protein you will get the amino acid phenylalanine. If, on the other hand, an AUU is present in the mRNA, you will get the amino acid isoleucine. Oops. So this is the codon in the RNA, and this is the amino acid in the protein. So as scientists, we use the genetic code to figure out what the protein sequence would be based on the mRNA sequence. You'll notice as you look at the genetic code in your textbook that there are more than one codon that code for the same amino acid. So leucine actually has 4 different amino acids that code for, 4 different codons that code for it. Notice that they all differ in the 3rd position. The first 2 positions are exactly the same. And you'll see that trend across here. Same thing with proline, same thing with serine. And that's just because of the way that the equipment works. You can have a little more wobble, is what it's called, in the 3rd position than you can in the other positions. A couple of other codons are really important. AUG has 2 jobs. AUG is always going to equal a methionine, which is an amino acid, but it also is going to mean start if it's near the beginning of an mRNA. So it's also called the start codon. There are also several stop codons. So notice that UAA, UAG, and UGA are all stop codons. That means that they signal the end of a protein. So a typical mRNA has a start codon near one end. Notice it's not exactly the end. This is a part that's not going to become a protein. So it's somewhere near the 5' end. There's that AUG. And then somewhere near the 3' end, again, not all the way to the end, but near it, is the stop codon. The part in between is the coding region. This is what will get made into protein or get read. So there's multiple codons all along this. Each codon will code for a specific amino acid that will create the protein. Another important part of this that I didn't mention yet is the ribosomal binding site. So in bacteria, they have a sequence here that the ribosome will bind to. In eukaryotes, there's the 5' cap that the ribosome will bind to. The start codon at the beginning of the mRNA is really important because it establishes something called the reading frame. So let me write a little sentence here for you. So as you're looking at this sentence, if I told you to start reading here and to make groups of 3, which is the way that the nucleotides are read, notice that I see the cat, the rat in the sentence. So if I wrote the same letters, so I'm writing the exact same letters, but instead I told you to start here and to start grouping your letters. Notice that even though it's the same exact letters, if you change the way that they're grouped, it no longer says the cat, the rat. So where you start is the reading frame. So the start codon, notice as I move along this sequence, there's a start codon. And the start codon AUG says, okay, group here. And now I create these groups. And that's going to give me my correct reading, the cat, the rat. If for some reason that gets shifted, So in this case, it's shifted because of a mutation. There's the addition of a U. Now, this is being grouped differently. Another way would be, what if I started here? So let me choose a different color here. What if I started grouping here? Now I'm getting very different combinations of letters. And so that where you start is the reading frame. If you don't start in the right position, then you have a reading frame shift. You're not going to get the right codons, and therefore you're not going to get the right amino acids. So again, in my little sentence here, if I started in the wrong place, I didn't see my words. So that's why reading frame is important, and that's why the correct start codon is really important. So DNA, we talked about when we're talking about transcription, is going to be read to make an mRNA, and the template strand is read. So in this case, the bottom one is the template strand. And remember, the way that I know that is that this is being made 5' to 3'. I'm moving towards the right. And so I must be reading this bottom one so that it's anti-parallel, because this is the 3' end. The RNA is always complementary to the DNA as well. So if there's a T in the DNA, then there's an A in the RNA. If there's a G in the DNA, there's a C in the RNA, and so on and so on. At some point along the RNA, there's a start codon, and where there's a start codon begins the reading frame. So now they'll be grouped in 3s from that position. This part is untranslated. It won't be made into protein. Eventually, there'll be a stop codon, which will stop the protein. Each one of these codons matches to a specific amino acid. So remember, we said that in the genetic code, AUG is coding for methionine. AGC is coding for serine. GAC is coding for aspartic acid. And CCC is proline, and so on and so forth. So the codons will match to a specific amino acid. That's genetic code. In order to make that match, the cell is going to use tRNAs. So tRNAs are also made out of nucleotides. And tRNAs have something called anticodons. So mRNAs have codons. tRNAs have anticodons. The anticodons match the codons. So for example, this is my start codon, 5' to 3' AUG. So my anticodon must be the opposite. UAC is antiparallel and complementary to the mRNA codon. These are going to match each other and help make the connection between the mRNA and the protein. And we'll see how that works in a later video. So we talked about this back in chapter 3, but I want to remind you that proteins have directionality too. We've been talking a lot about the directionality of RNA and DNA. So we've talked about how there's the 5' and 3' end, for example, here. The 5' end of the mRNA is going to code for the N terminus of the protein. And the 3' end of the mRNA correlates to the C terminus of the protein. So here's AUG. So that means methionine is at the N terminus. Then there's a serine, then there's a valine, a tyrosine, a cysteine, and then I have my C terminus. So just like we always put 5' and 3' on our RNA and DNA, we also need to put N terminus and C terminus on our polypeptide and protein sequences. Okay, I want to work through this practice problem with you. So you know that a template strand of DNA is 3' and there's the sequence here. I always break my sequences into groups of 3 to make them easier to read. And the first thing I need to do is to make an mRNA from this. So my mRNA needs to be antiparallel, and then it needs to be complementary. So I'm making my base pairs. Remember, if it's RNA, there's U's, not T's. CHE, CAU. So there's my RNA sequence, and again, it's antiparallel and complementary. I have base pairs there. Now I'm going to read it in these groups of 3. And so my first codon is AAA, so I go to my genetic code. There's AAA, there's AAA. And this is my N terminus. Whoops. This is my N terminus here. GCC, so GCC, UUU, CAG, and last codon here is CAU, CAU. So that's my protein sequence that comes from this template strand. So sometimes people ask me if they have to memorize the genetic code. No, you do not have to memorize the genetic code, but expect to do problems like this on the exams and on the lab practicals. And in that case, the genetic code would be provided for you. So you'll see this chart on exams. You don't have to have it memorized.